That frozen dinner on your plate isn't just convenient, it's already partially digested. The processing plant has done what your body evolved over millions of years: break down proteins into amino acids, convert complex starches into simple sugars, separate nutrients from fiber. Every bite has been pre-programmed for rapid absorption. When they say processed food, they never tell you what that really means. That your body's ancient digestive rhythms have been outsourced to machines. That industrial enzymes and mechanical forces are mimicking the chemistry that should happen in your gut. The evidence is hidden in plain sight, in the instant dissolution of processed cereals, in the hyper-palatable textures engineered for immediate pleasure, in the rapid sugar spikes that follow each artificially refined bite. This isn't just food made convenient, it's digestion itself, industrialized. On this episode of Food Truth, we're breaking open the science of food processing to reveal how factories aren't just preparing your food, they're pre-eating it. We'll explore the biochemistry that transforms whole foods into their processed counterparts and what happens when machines start doing the job your body evolved to perform. Join us as we uncover the fascinating and unsettling reality of processed foods and why convenience comes at a price your metabolism never expected to pay. This is Food Truth Episode 6, The Processing Show. Take two identical wheat berries, hold them in your palm, feel their hard outer shells, the protective layers that have kept their nutrients safe for months. Now place one on your tongue. Your saliva begins to soften it, the first step in a process that evolved over millions of years. Now listen to the industrial grain mill crushing its twin, steel rollers operating with precision force, accomplishing things in seconds that would take your body hours. Your stomach produces hydrochloric acid to break down food. That same acid is now spraying in industrial vats, pre-digesting proteins in that protein shake powder. Touch your abdomen. Feel the gentle waves of peristalsis moving food through your digestive tract. Now listen to the mechanical homogenizer, forcing particles through microscopic holes under extreme pressure, shattering cell walls your body would have to work to break down. Place a piece of cheese on your tongue. Your body releases lipase to break down its fats, protease to dismantle its proteins. In the factory, those same enzymes are being produced in vats of engineered bacteria, concentrated and purified to levels your body never evolved to handle. What takes your digestive system hours happens in minutes. Your small intestine cells carefully select which molecules to absorb, using carrier proteins evolved over millennia. Industrial ultrafiltration membranes now mimic this selectivity, separating molecules by size and charge. Touch the inside of your cheek. Feel the delicate membrane that decides what enters your body. Now imagine industrial filters operating at pressures that would tear your natural membranes apart. Feel your liver processing nutrients, releasing precise amounts of enzymes to break down just what's needed. Industrial processors don't wait. They pre-digest, pre-absorb, pre-process. They bypass your body's careful timing, its evolved wisdom about what should be broken down and when. This is the truth about processed food. It's not just convenient. It's partially digested before you even take your first bite. Every crunch of that breakfast cereal, every smooth spoonful of that protein shake, every bite of that energy bar tells a story of digestion outsourced to machines. Your body's ancient wisdom has been replaced by industrial efficiency. The transformation of whole foods into processed counterparts begins with mechanical force, both in your mouth and in the factory. Industrial processing has found ways to replicate and intensify the mechanical forces of chewing, creating pressures and shear forces that your body could never generate. Understanding these forces reveals how processing fundamentally alters food structure. High-pressure homogenization can generate pressures exceeding 20,000 psi, that's 20,000 pounds per square inch, forcing food particles through microscopic gaps that can be as small as 1/10 of a micrometer. It's extremely small. For comparison, your molars generate pressures of only about 150 psi when chewing. This massive difference in force doesn't just break down food faster, it creates entirely new physical structures that your body never evolved to process. Roller mills in grain processing use differential speeds to create both compressive and shear forces. While your teeth crush and grind, These mills can separate grain components with microscopic precision. The endosperm, bran, and germ can be fractioned into distinct streams, allowing for the creation of refined flours with properties that would be impossible to achieve through traditional milling. Extrusion processing subjects food to a complex combination of pressure, heat, and mechanical shear. Materials are forced through dies under pressures that can exceed 1,000 psi while being subjected to temperatures above 150 degrees Celsius. This combination of forces and heat can gelatinize starches and denature proteins in milliseconds, a process that would take your digestive system hours. Ultrasound processing uses high-frequency sound waves to create cavitation bubbles in liquid foods. When these bubbles collapse, they generate localized spots of extremely high pressure and temperature, up to 5,000 Kelvin and 1,000 atmospheres. These microscopic hotspots can break cellular structures and modify food components in ways that digestive forces never could imagine. The particle size reduction achieved through industrial processing often creates surface areas hundreds of times larger than what mechanical digestion produces. A single gram of microfluidized protein can have a surface area exceeding 30 square meters. This massive increase in surface area dramatically changes how these particles interact with digestive enzymes. High-pressure processing can subject foods to pressures up to 87,000 psi, forces that can reshape proteins and modify cell membranes without generating heat. These pressures are transmitted instantly and uniformly throughout the food, affecting structures in ways that are fundamentally different from the gradual breakdown of digestion. The speed of industrial mechanical processing introduces another crucial difference. While digestion gradually breaks down food structures over hours, allowing for controlled release of nutrients, industrial processing can shatter these structures in seconds. This difference in timing can affect everything from nutrient absorption rates to satiety signaling. Modern processing equipment has also achieved precise control over particle size distributions that would be impossible in natural digestion. This allows for the creation of products with specific textural properties and mouthfeel, often designed to bypass natural satiety mechanisms that depend on mechanical breakdown during chewing. These mechanical forces don't just break down food, they fundamentally alter its behavior in your digestive system. Pre-broken cellular structures can release nutrients more rapidly. Changed particle sizes can affect gut transit time, and modified textures can alter the very way your body responds to these foods. Chemical processing in food manufacturing mimics your digestive chemistry while operating at extremes your body never evolved to produce. Where your stomach maintains a pH between 1.5 and 3.5, industrial processes can utilize pH levels across the entire spectrum, from highly caustic pH 14 to extraordinarily acidic pH 0, creating chemical environments that would destroy living tissue. Protein modification through acid hydrolysis subjects food proteins to concentrated hydrochloric acid at elevated temperatures, often exceeding 100 degrees Celsius. This aggressive treatment can break peptide bonds in minutes that would take your digestive system hours to process. The resulting hydrosalates contain peptide fragments in patterns distinctly different from those produced by natural digestion. Alkaline treatment of foods, particularly in corn processing, nixtamalization, uses calcium hydroxide at pH levels above 11. While this traditional process improves nutrient availability, modern industrial versions operate at higher concentrations and temperatures than traditional methods. These intense conditions not only modify proteins, but can create novel compounds not found in natural digestion. Industrial enzymes, often produced by genetically modified microorganisms, can achieve concentrations hundreds of times higher than those in your digestive system. These purified enzymes can break specific chemical bonds with precision that digestive enzymes never evolved to match. A single industrial enzyme preparation can process more protein in an hour than your body could digest in a day. Oxidative processes in food manufacturing introduce chemical modifications that have no parallel in digestion. Treatments with hydrogen peroxide, ozone, or other oxidizing agents can modify proteins, break down complex carbohydrates, and alter fat molecules in ways that create entirely new chemical structures. These modifications can affect both nutrient availability and the formation of potentially problematic compounds. Chemical extraction processes use solvents that would be toxic to living systems. Hexane extraction of oils, for example, can achieve near complete separation of lipids from plant materials, a level of extraction that your digestive system could never approach. These processes create highly purified ingredients, but can also strip away beneficial compounds that normally accompany these nutrients. The speed of industrial chemical processing introduces another crucial difference. While your digestion gradually exposes food to changing chemical environments as it moves through your digestive tract, industrial processing can subject foods to extreme chemical conditions instantaneously. This rapid treatment can create reaction products that would never form during natural digestion. Chemical modification of food components, such as the hydrogenation of oils or the phosphorylation of starches, creates molecular structures that your digestive system never evolved to process. These modified ingredients can behave differently in your body, affecting everything from absorption rates to metabolic responses. The combination of extreme pH, temperature, and pressure in industrial processing can create Maillard reaction products and other chemical compounds at rates and concentrations impossible in natural digestion. While some of these compounds contribute to desired flavors and colors, others may represent entirely novel chemical structures from an evolutionary perspective. Quality control in industrial chemical processing focuses on specific endpoints: protein solubility, degree of hydrolysis, or extraction efficiency, rather than the broader metabolic compatibility that natural digestion evolved to achieve. This targeted approach can create ingredients that meet technical specifications while potentially disrupting the complex chemical sensing systems that regulate digestion. While your digestive system produces a carefully orchestrated sequence of enzymes, industrial processing deploys engineered enzymes at concentrations and specificities that dwarf their natural counterparts. These industrial enzymes, often produced by genetically modified microorganisms, can achieve reaction rates up to 1,000 times faster than digestive enzymes, fundamentally altering how food components are broken down. Modern enzyme engineering has created variants that operate under conditions that would destroy their natural counterparts. Where digestive proteases work best at body temperature and specific pH ranges, industrial proteases can function at temperatures exceeding 90 degrees Celsius and pH levels from 4 to 12. This environmental tolerance allows for processing conditions that create different breakdown patterns than natural digestion. The specificity of industrial enzymes has been pushed beyond natural limits. While pancreatic amylase breaks down starch somewhat randomly, engineered amylases can target specific bond types with remarkable precision. This selective breakdown creates sugar profiles and chain lengths that your digestive system would never naturally produce, affecting everything from sweetness perception to absorption rates. Concentration makes another crucial difference. Your pancreas carefully regulates enzyme secretion based on meal composition and metabolic needs. Industrial processes can use enzyme concentrations of hundreds of times, creating reaction conditions that bypass these natural regulatory mechanisms. A protein shake might encounter more protease activity in production than it would during an entire digestive cycle. Industrial enzymes often work in synergy with multiple specialized enzymes attacking different components simultaneously. While your digestion is sequential, salivary amylase, then pepsin, then pancreatic enzymes, industrial processing can deploy complex enzyme cocktails that break down multiple components at once, creating novel intermediate products. The purity of industrial enzymes introduces another significant difference. Digestive enzymes work within a complex biological matrix that includes inhibitors, activators, and modulators. Industrial enzymes, purified and optimized for specific reactions, operate without these natural checks and balances, potentially creating product profiles that differ significantly from natural digestion. Timescales in industrial enzymatic processing bear little resemblance to natural digestion. What would take hours in your digestive tract can happen in minutes or even seconds in industrial processes. This rapid breakdown can create different metabolic responses when these pre-digested components enter your system, potentially overwhelming normal absorption and processing mechanisms. Enzyme immobilization technologies, unknown in natural digestion, allow industrial processes to reuse enzymes multiple times while operating under more extreme conditions. These immobilized enzymes can create consistent breakdown patterns that differ from the variable, responsive nature of digestive enzymes that adapt to meal composition and metabolic needs. The stability of industrial enzymes has been engineered to exceed biological limits. While digestive enzymes are constantly produced and broken down, industrial enzymes can maintain activity for weeks or months. This extended stability allows for processing approaches that would be impossible in biological systems, creating foods with degrees of pre-digestion that nature never intended. Quality control in enzymatic processing focuses on endpoint measures like degree of hydrolysis or specific sugar profiles, rather than the holistic regulation seen in digestion. This targeted approach can create ingredients that meet technical specifications while potentially disrupting the complex signaling systems that evolved to regulate digestion and metabolism. Your body evolved sophisticated systems to regulate food intake, carefully balancing energy consumption with metabolic needs. But processed foods bypass these ancient regulatory mechanisms in multiple ways, creating a perfect storm for weight gain. The very efficiency of industrial processing becomes a metabolic liability when it encounters systems designed for whole foods. The speed of nutrient absorption makes a crucial difference. When industrial processing pre-breaks food components into their simplest forms, it shortcuts the gradual digestion process that normally regulates nutrient release. A processed carbohydrate can flood your bloodstream with glucose in minutes, triggering an insulin response that natural foods would create over hours. This rapid absorption disrupts normal satiety signaling. Texture modification eliminates important satiety cues. Your body evolved to gauge food intake partly through mechanical effort, the work of chewing and breaking down food structure. Processed foods, with their carefully engineered textures, often require minimal chewing and breakdown. This bypasses the mechanical signals that would normally help regulate portion size and meal duration. The concentration of calories in processed foods often exceeds anything found in nature. While natural foods contain calories bundled with fiber, water, and complex cellular structures, processing can create energy densities that your satiety mechanisms never evolved to handle. A single bite of a processed snack can deliver the same calories as several bites of its whole food counterpart. Processing can separate nutrients that naturally occur together, disrupting the complex interplay of signals that regulate appetite. Fiber, which normally slows digestion and promotes fullness, is often removed. Natural food matrices that would pace nutrient absorption are broken down. This separation means your body receives individual components without their natural moderating elements. The combination of processed fats and carbohydrates creates reward responses that exceed those of natural foods. While natural foods typically emphasize either fats or carbohydrates, processed foods can combine them in ratios that trigger excessive dopamine release, promoting overconsumption through enhanced food reward pathways. Industrial processing can create particle sizes that affect both absorption and satiety. Ultrafine grinding produces particles small enough to bypass normal digestive barriers, while carefully engineered textures can affect how long food stays in your stomach. These physical modifications can decrease the time between eating and hunger signals. The removal of bitter compounds and other natural flavor components eliminates important satiety signals. Your body evolved to use subtle taste cues to regulate intake, but processing often strips these away, replacing them with engineered flavors that encourage continued consumption without providing the same metabolic feedback. The protein to energy ratio in many processed foods falls below what your body expects from natural foods. This ratio is a key signal for satiety. Even when it's disrupted, you may need to consume more total calories to reach the protein levels that triggers fullness. This protein leverage effect can significantly lead to increased overall energy intake. The temperature and lubricity modifications in processed foods can bypass oral exposure time, another important satiety signal. Foods designed to dissolve quickly or slide down easily reduce the time your oral sensors have to gauge energy density and nutrient content, leading to delayed satiety signals and potential overconsumption. Your brain's reward system evolved to encourage behaviors essential for survival, with food reward being one of its most fundamental functions. But processed foods can trigger these ancient circuits with unprecedented intensity, creating reward responses that natural foods could never generate. This isn't just about pleasure, it's about how industrial processing creates stimuli that overwhelm normal reward regulation. The dopamine response to processed foods can be up to twice as strong as that triggered by their unprocessed counterparts. While natural foods gradually release their components during digestion, processed foods can deliver concentrated rewards to your reward circuits almost immediately. This rapid delivery closely resembles the dopamine spike patterns as seen with other rewarding and illicit substances. The combination of fat and sugar in processed foods creates a supernormal stimulus, one that exceeds the parameters your reward system evolved to handle. In nature, foods high in fat rarely contain high sugar levels, and vice versa. But processing can combine these in ratios that trigger excessive reward responses, activating multiple pleasure pathways simultaneously. Texture and mouthfeel in processed foods are engineered to maximize oral reward. Natural foods require chewing, which gives your brain time to process reward signals. But many processed foods are designed to dissolve rapidly or provide immediate texture rewards, creating intense pleasure signals before satiety mechanisms can activate. The brain's opioid system, which responds to pleasurable experiences, shows enhanced activation with highly processed foods. The rapid availability of refined carbohydrates and modified fats can trigger the release of endogenous opioids more intensely than natural foods, potentially creating subtle but significant conditioning over time. Flavor enhancement through processing can stimulate the reward system in ways that natural flavors cannot match. While natural food flavors are complex and subtle, processed foods often contain concentrated flavor compounds that provide immediate, intense reward signals. This can reset your pleasure threshold, making natural foods seem less rewarding by comparison. The predictability of processed foods plays a crucial role in their reward impact. Natural foods vary in flavor and texture, but processed foods deliver precisely engineered sensory experiences every time. This consistency can strengthen reward learning, as your brain knows exactly what pleasure to expect with each encounter. Processing can create novel food properties that exploit the brain's reward prediction error system. When a food delivers more reward than expected, through carefully engineered flavor releases or texture transitions, it can amplify the learning that drives future cravings. This mechanism helps explain why certain processed foods become particularly compelling. The speed of reward delivery matters enormously. Your reward system evolved to handle the gradual pleasure of eating and digesting natural foods. But processed foods can deliver their entire reward load in seconds, creating abbreviated but intense pleasure spikes that can be particularly reinforcing to reward circuits. The combination of sensory rewards and processed foods often targets multiple neural pathways simultaneously. Dopamine, opioid, and serotonin systems can all be activated by a single bite. This concurrent stimulation of multiple reward pathways creates a more compelling experience than natural foods, which typically engage these systems more modestly and sequentially. Industrial food processing has fundamentally altered the relationship between human biology and food in ways that no other technological advancement has achieved. This isn't just about changing what we eat. It's about creating food forms that interact with our bodies in ways that bypass millions of years of evolutionary programming. The convergence of mechanical, chemical, and enzymatic processing creates foods that exist outside our evolutionary experience. When industrial forces can pre-digest, pre-absorb, and pre-process nutrients, they circumvent the very digestive processes that help regulate our metabolism. A single processed food product might combine multiple engineered components, each designed to maximize efficiency at the expense of biological appropriateness. The speed of nutrient delivery from processed foods creates metabolic responses that natural foods never could. While your body evolved to handle the gradual release of nutrients from whole foods, processed foods can flood your system with pre-broken components in minutes. This rapid delivery affects everything from insulin response to satiety signaling, creating metabolic patterns that can overwhelm normal regulatory systems. Our sensory systems, designed to evaluate and respond to natural foods, encounter engineered stimuli that exceed their normal operating parameters. Processed foods can combine flavors, textures, and nutrient densities that send amplified signals to our brain's reward centers. These supernormal stimuli can reset our pleasure thresholds and alter our food preferences at a neurobiological level. The removal of food structure through processing eliminates important signals that our digestive system uses to pace and regulate intake. Natural cellular barriers, fiber matrices, and complex nutrient arrangements that would normally slow digestion and absorption are stripped away. This structural simplification means our bodies must rely on fewer cues to regulate food intake and metabolism. Processing can separate nutrients that naturally occur together, disrupting the synergistic effects that whole foods provide. When industrial methods isolate and concentrate individual components, they break the natural ratios and combinations that our metabolic systems expect. This separation can affect everything from nutrient absorption to hormonal signaling. Time itself becomes a critical factor in processed foods' impact on health. The industrial ability to transform food structure in seconds rather than the hours of natural digestion creates temporal mismatches between nutrient availability and metabolic processing. Our bodies simply haven't evolved mechanisms to handle such rapid delivery of concentrated nutrients. The scale of processing introduces another dimension of biological mismatch. Industrial methods can create uniform particles, concentrated nutrients, and engineered textures at levels that would be impossible to achieve through natural food preparation. This standardization eliminates the variability that our digestive and sensory systems evolved to handle and interpret. Modern food processing represents a fundamental shift in how humans interact with food at a biological level. It's not just that processed foods are more convenient or longer lasting. They create entirely new patterns of interaction between food and human biology. Understanding these patterns is crucial for comprehending the health challenges of our modern food environment. The future of food processing needs to balance efficiency with biological appropriateness. As we understand more about how processing affects human health, the challenge becomes creating processed foods that respect our evolutionary heritage while meeting modern needs. This might mean developing new processing methods that preserve or mimic the important signaling properties of natural foods while maintaining the convenience of processed products. Thanks for joining me on this episode of Food Truth and taking the deep dive into processed foods. I hope you learned a thing or two. If you want more Food Truth, go to www.getfoodtruth.com. Please join us next time as we take the next deep dive into the world of micronutrients. We'll see you then.